everyone, my name is Alexandra and I'm excited to present to you my summer research project which was conducted here in the Levinson lab. This summer I worked on RNAi optimization for ferritin gene knockdown in Colorado potato beetles. But before we jump into the science, let me introduce you to one of the beetles. While the beetles may be cute, they are actually a major agricultural pest, wreaking havoc on crops all over the world. One of the ways they are able to be such a persistent pest is because they undergo diapause during the winter, which is a pre-programmed state of dormancy. During diapause, the beetles suppress their metabolism by breaking down their flight muscle and degrading all the mitochondria it contains. This process, called mitophagy, releases excess free iron into the cell, which can trigger ferroptosis. Ferroptosis is an iron-dependent form of cell death, which happens when extra iron combines with oxygen to create reactive oxygen species, also known as ROS, that can cause extreme damage to the cell. But here's the thing. Even though these beetles seemingly have the perfect conditions for ferroptosis to occur, they do not die. They continue living on happy as can be. So the question becomes, why is that? This may be due to a gene that encodes for a protein called ferritin. Ferritin works to sequester iron, and because of this, ferritin might have the protective properties that helps Colorado potato beetles survive an otherwise lethal fate. Hence, my hypothesis was that by optimizing RNAi technology targeting the ferritin gene, I can achieve a successful knockdown, thereby creating a tool I can then use to study ferroptosis further during my thesis this year. RNAi is a transient gene silencing technique where you can actually silence a gene, or knock it down, by introducing double-stranded RNA that targets your gene of interest. To do this, I synthesized double-stranded RNA targeting the ferritin gene by first optimizing a PCR template containing the DNA sequence of ferritin. I tested several cycling temperatures and found that 62 degrees produced a clear band at the expected size during gel electrophoresis, a technique that helps you visualize the product, confirming successful amplification. This optimized PCR template was then used to synthesize double-stranded RNA, which was also subjected to a quality check on a gel. With the correct synthesis being confirmed, the double-stranded RNA was ready for beetle injections, which included two treatment groups. One group was injected with double-stranded ferritin and dissected either 3 or 6 days post-injection, and the control group, injected with phosphate-buffered saline, was also post 3 or 6 days. During the dissection, I collected flight muscle that was then subjected to RNA extraction. This RNA was then used to make cDNA, which is necessary for quantification using quantitative PCR or qPCR, which is a technique that measures the abundance of a specific DNA molecule, and in our case, it is ferritin. So after doing all of this, here are the results I found. Success! Ferritin abundance was knocked down after 3 days. So as you can see, the y-axis shows relative normalized ferritin abundance, and the x-axis shows days after double-stranded RNA injection. We see that in comparison to the beetles treated with PBS, the amount of ferritin has definitely decreased, indicating a successful knockdown. But now you might be wondering, why does ferritin abundance increase 6 days post-injection relative to the control? Here's what I think might be happening. Since ferritin is an essential gene, it may be that after knockdown, the panicked beetles purposely increase the production of ferritin, leading to the overcompensation observed. For my thesis project this year, the experiment will be repeated in diapausing beetles, and I will also measure iron concentration and lipid peroxidation, which are classic characteristics of ferroptosis. Based on my summer results, I am also considering additional RNAi injections to make sure ferritin remains knocked down in the day 6 beetles. I am passionate about continuing this project because silencing the ferritin gene and observing its effects can deepen our understanding of the beetle's unique cellular processes. This insight could help develop better strategies to manage their destructive agricultural impact, such as with the development of RNAi pesticides. Additionally, studying ferroptosis in these beetles can help illuminate its role in a vast array of human diseases, potentially guiding the efforts to mitigate these illnesses through ferroptosis manipulation. Thank you.